Welcome to this episode of Eureka, and this is also a Bhatnagar Award special episode. As you all know, the Shanti Swarup Bhatnagar Award is the highest award for scientific research given in India. 2020 awards have been declared, and the guest we have today, Dr.Subhadeep Chatterjee, from Center for DNA Fingerprinting and Diagnostics, a DBT lab from Hyderabad, is a recipient of this award for the year 2020. Sir, thank you for being with us. It's a very, we are very honored for your presence. And first, please accept our congratulations for the award that you got for this year, 2020, for your phenomenal work on how the bacteria and some of the very important agricultural crops interact. I'm sure your work is going to have a very important impact on the way we produce food in future, when we want to avoid pesticides, when we want to go for more organic way, use of science and technology to create higher productivity without necessarily using environmental destruction chemicals. I'm sure these are all something that we are going to discuss in the show. So first, let me welcome you to the show, and then let's start with our discussion. See, your work is what is called as bacterial quorum sensing. That's what has been mainly mentioned as one of your key area of research. So can we understand what is this quorum sensing?
See, in all forms of life, communication is very important. So for response, so communication from the lowest organism to the highest human being language evolution is very important for survival. So from dancing bees, to the firefly blinking, all are forms of communication. So this communication system, animals and all the living organisms have evolved to better respond to the environmental changes. So they tell to their fellow mates what is coming up. Whether there is a danger, whether they have to escape, whether there is a food. If there are beautiful flowers in the field, the honeybees communicate with the other members that there is good nectar.
Like particularly honeybees tell that nectar-bearing fruit, I mean, flowers are on this direction, this distance, that kind of information they communicate to other bees, right?
That's called a dancing language of honeybee. So when this microbiology field started, so this from long ago, so people have been studying bacteria, but nobody thought of that bacteria can communicate with each other.
I see. So you are saying that like bees communicate with each other, or ants communicate with each other, the bacteria also communicate with each other.
Bacteria can also communicate with each other.
Very interesting.
So it came up in a very surprising experiment, which will be very good for, is a very general our knowledge, that people actually indirectly got that answer. So, the people were studying marine squid called as Hawaiian squid, which is found in Pacific. So it has a very nice light organs. It emits light in the night. And marine biologists tell that this light actually helps the squid to get its food in the night. And in the daytime, it sheds off the light. So when people studied in detail, they found out that light produced by this squid is not its own light. It is by a group of bacteria which colonizes the organs which are called light organs of this squid. So now then people started interested how this bacteria is producing light. They cultured that bacteria in the lab and found that this bacteria only produces light when they are in high number.
I see. So when they are alone, they don't produce any light.
They don't produce. Or when they are less in number, they don't produce. Only when they are in high number, they will produce.
Okay.
So then people thought maybe this high number of bacteria is secreting something
Uh-huh.
in the medium which is making the communication possible.
Okay.
So they did a very clever experiment. They took the spent media from the high density bacteria and put it in those bacteria which are not talking.
Okay.
And then the non-talking bacteria started to talk. That was the first proof that bacteria has some message or some signals which has been made in high number, which makes them to communicate with each other.
Okay.
Then the whole field of social microbiology evolved. And in late 1990s.
See, common knowledge, something that we study in schools when we study biology or zoology is that there are something called social animals, ants or bees. They have communication, but then animals which are alone may not have that kind of a sophisticated
advantage.
But now you are saying that bacteria, which are like single cell organisms.
Single cell, unicellular, solitary living, lonely, people have thought of. But now people are finding it's a completely different scenario. They talk with each other with different signaling molecules. They talk in group. They talk with each other also. Like one species of bacteria can talk with other species of bacteria.
Okay.
And this talking is happening always in the nature to regulate their social behavior.
Okay. So now let's come to the wording that we started with. I mean, that's how we started our discussion, that quorum sensing. Quorum, for example, many of us would know that in a meeting, the minimum number of people who should be present so that the meeting can be conducted. So suppose if you have a committee, there is a quorum for the committee.
Same.
If people come lesser than that number, then you have to defer that meeting. Like, for example,
voting or
right. So you are looking at minimum number of bacteria which should be available to conduct
a behavior. So this is literally the meaning of quorum sensing.
That's a sense of quorum, right?
Quorum.
So people also say that, for example, the bacteria forms thin films. Which is then it is not like a single cell, but it's acting like a. So that is also basically because of this kind of quorum sensing. Can you explain to us about that?
So when people started looking more closely the bacteria in natural environment, so they found like film-like structures which are coming up in your operation devices, in cathedra, in pipelines, in your shower, bathroom shower. So if you open your bathroom shower, you will get a film of this bacteria. Even a lot of diseases like cystic fibrosis, Pseudomonas aeruginosa colonizes your lungs and it forms a film-like structure, and that complicates the matter. So these are film-like structure when people started to study, and even in our lab also we study. So we found there is actually bacterial big housing society. So they have big buildings. They have clubhouse. Like that. They have canals. To pass off the nutrients, drainage. So it is actually whole thing is made up of bacteria and the products which is secreted by the bacteria for the bacteria to protect it from the hostile environment. And also to organize to do certain task which will be beneficial when you are in high in number.
I see.
When you are low in number, it is very similar to, I will give you an example. Suppose there is a barrier. Now there is a single person who is firing a bullet to break the barrier. So the bullets are so small and there is firing power is so small because one person, the barrier may not break. But if there are 1,000 people firing at the same time, there is a good chance that the barrier will break. So this is the whole concept of quorum sensing in a layman's language. There is always group benefits to perform a social task in unison. But if you are doing the same job alone, you may do the job, but you will not get the benefit. Suppose you spend 100 rupees he will not expected to get 200 rupees. But if you are doing in a group, you are spending 100 rupees, the return may be 1000 rupees.
Okay.
So that
So,
is
if I can think like this, for example, when the fish don't swim alone, they form a school and swim.
Yes.
Then they have a particular pattern which is like aerodynamic.
Sure.
So when they are actually swimming as a group, many of the fishes may not have to expend the same kind of energy that if they are going to swim alone. If you average out the total energy, it will be much less than the individual fish which you would have to swim. I mean, if they have to spend if they have to swim, right? Sure. So actually together they benefit if they actually act like a group.
Actually swim together. And there is another angle to your example. They actually escape the predator if they swim together. Okay. So they give an appearance of a big fish.
Okay.
So there is an advantage in doing.
So there is a huge advantage being united than disunited.
Than unicellular.
That's a very interesting.
But I will tell you, there are advantages as well as disadvantages.
Yeah.
Okay. And then I will go to my own research work, and then so this quorum sensing I was talking about. So this thing developed, and then people started to look in more detail, and they found out it is an amazing field.
Okay.
So different bacteria is communicating with different languages. And many bacteria, they don't speak, but they can listen.
I see.
So they have receptors. They can listen to their neighborhood.
I see.
So it is very similar to your noisy neighborhood joke. Okay. Like the neighbors are more interested who has come to their neighborhood rather than the person who has come. So they have ears or receptors which can listen to the languages of others so that they can change their behavior based on their neighbors.
Okay.
So this complication also happens. The other complication in quorum sensing complexity is that some bacteria speak in multiple languages.
I see. Okay.
So for example, Vibrio cholera, which causes a very bad disease called cholera, so it can speak in 2 languages.
I see.
Pseudomonas aeruginosa, which causes cystic fibrosis, it can speak in 3-4 languages.
Okay. That's a very interesting.
And another interesting analogy to this language is that it is very similar to our Indian language.
Okay.
You see, we have a Sanskrit as a base. Many bacteria, one family of signaling molecule is acyl homoserine lactone. So lactone is the base, and they vary their acyl chain very similar to Indian languages like Hindi, Bhojpuri. So different chain lengths are added to have different dialects.
So for example, like we started with the discussion of honeybee, which kind of does a dance to communicate with others. But whereas when we are coming about bacteria, it emits certain chemical which acts like signals, and the other bacteria is able to respond to that chemiosensing, and then that essentially acts like a communication medium. That's essentially the point that we are getting it, right? So what we'll do is that we'll take a very short break here.
Yeah, sure.
Then we'll continue this conversation. Sure. Keep watching Eureka. We'll take a very short break. This conversation is really interesting. Like you and I speak language, the bacteria speak language, and then act unitedly. We are going to talk about how and what he has worked on particularly the agricultural crops after the break. Keep watching Eureka.
Watch India Fights Back with me, Vishal Dahiya, at 7:30 p.m. only on Dr. Subhadeep Television.
Welcome back to Eureka. We are having a very wonderful conversation with Dr. Subhadeep Bhattacharya. He is with the Center for DNA Fingerprinting and Diagnostics, Department of Biotechnology Institute in Hyderabad, and he has been awarded the India's very prestigious scientific award, Shanti Swarup Bhatnagar Award for 2020. and he is working on what is called as quorum sensing by bacteria, which in, let's say, ordinary person's language, we can say that how bacteria talk to each other, particularly when they are in large numbers. Sir, we before the break talked about in general about quorum sensing and how bacteria communicate with each other. What are all the various facets of it? Now we are very eager to know about your work. Your work is essentially on some of the essential agricultural crops and how they interact with bacteria which destroy them. I mean, are interacting with them.
Sure,
so, can you explain to us what is the work?
Yeah, sure. So we work on a group of pathogens called as Xanthomonas.
Okay.
So Xanthomonas is one of the most diverse group of plant pathogens, which causes very economically important diseases in important economic crops like rice, vegetables, even fruits, cabbage, like citrus, walnuts, everything. So particularly I focused our work mostly on the rice and the vegetable pathogen. And rice, why? Because it's a major crop in India, and particularly in the southern state, it is heavily grown. And the problem starts when people started growing hybrid rice. So hybrid rice is very prone to Xanthomonas infection. It causes a very dangerous disease called a bacterial leaf blight.
I see.
So when it happens, the Xanthomonas completely dries up the rice and kills the rice. So when we started working on this bacterial system, we discovered a quorum sensing system in this bacteria. Which is very unusual than the other bacteria, that it is not by acyl homoserine lactone. It is by a long chain fatty acid.
I see.
We call as diffusible signal factor. We named it as DSF. Diffusible signal factor.
So if again a common person's language, we can say that the DSF is the language of this particular pathogen.
This particular bacteria, Xanthomonas.
Very nice.
So then we started working more on the molecular aspect, how this signal is made, how the bacteria is sensing, and what does it regulates. So after a lot of research work, we figured out the signal system in this bacteria, how it perceives the signal, and how it does the work based on the signal input. So in a nutshell, in a take-home message, what we got is that we found out this signaling molecule is very important to trigger biofilm formation inside the plant, inside the host. And this biofilm formation is very important. But it is a two-way process. So at low signal, that means low language communication, it also gives a signal to the bacteria to move inside the host. So low signal is a trigger for the bacteria to find its home. That is the plants. Once it enters inside the plant, the signal starts building up as the bacteria grows. And then it tells the bacteria, now you are inside. You form a biofilm and destroy the host. So it is a two coin, same language, interpreted in a different way. When in low in amount, it tells the bacteria to attack. When in high in amount, it tells the bacteria you form your home inside.
So it is something like, for example, a mother, when she is happy about you, calls you your name in a particular way. When she is angry about you, says the same name in a different way. We understand that she is happy or angry. Right?
Very, very similar to that. And what we found out when, that means when the signal is low, it activates its motility and virulence factors. Virulence factors are those secreted by the bacteria to cause disease. And at high cell density, it makes a lot of these attachment proteins in the bacteria to grip on the host cell wall. So, and it is a xylem colonizing bacteria. Xylem is very similar to blood vessels in higher animal, where the water is drawn from the ground to
and then the nutrients, etc., go through that.
And this bacteria chooses only that place. to get the nutrients and to spread. It's an excellent way of spreading in the plant.
Okay.
So we discovered that it makes this attachment proteins and virulence vector in a very orchestral manner. So first it makes X, suppose a factor, then only Y, and when the signal molecule reaches a threshold, it makes XYZ.
Okay.
So it is a very orchestra way of performing your act in a dramatic way.
Okay.
Because if you first show your villain, then the show is over, right?
Yeah, yes.
So it is very clever that it utilizes the language molecule to keep its weapons hiding at the initial stage, so that the plant will not recognize it. But the weapons are required for destruction, for getting food, ultimately. So it only unleashes the weapons when they are enough in number. So that when the plant mounts a defense response, even if it kills 50% of them, the other 50 will survive.
Okay.
That is the strategy.
So if they had started actually attacking the plant in the earlier phase itself, the bacteria would have been wiped out, not the plant, right? I mean,
this is the way we have figured out. It utilizes the quorum sensing system to its own benefit to hide those weapons, to take out those weapons which are not that alarming to the host at the initial stage. Only take out your weapons and the factors when you need the food and when there is no remedy. It is very similar to robbing a bank. Suppose one robber goes, it can be overpowered by the bank security.
Yeah.
But if multiple robbers flash their weapon, then the bank has to surrender, right?
This is a kind of thing.
This is the concept, yeah.
Okay.
So now I will tell you a little bit. So in this game of language and this thing, we discovered another aspect of quorum sensing. This is for the first time in the world.
Okay.
Is what is called as reversible heterogeneity.
Yes, that's what I wanted to ask you about. Yeah, please.
Yeah. So this is a new concept which has come up, which is called a bet hedging. I will give you a very simple example. For example, in your home, there are 4 members, right? So one member is earning, one member is cooking, another member is helping in the household job. The 4th member doesn't do anything. Okay. He enjoys the benefit of those 3, but he is very good in computers. Okay. Now suppose there is a situation like Corona, everybody is working online. And the bread earner also has to work online. Now his computer fails. Now this 4th fellow who has not contributed much to the family chips in and helps in setting up his computer thing. So in the indirectly way, there is a cheater in the family, but cheater has been taking the benefit of the performers, but in some environmental condition or some condition, some benefit it will give it to the.
It's also coming to the
family or the community. Very similar to these bacteria cheats. So what we discovered, 80% of the population in this bacteria communicates and perform the task, quorum sensing. 20% of them remain silent.
Like this 4th person in the house.
Yeah, just keep quiet. So what this 20% do, they take initially at the initial infection stage the benefit of the performers. But once the biofilm is formed at high cell density, imagine a home in which too much members are packed off. Soon your ration will be limiting. You have to go to the other place to get the food. Now who will go? Everybody is tuned to staying in that home because of the quorum sensing.
Yeah.
Now this 20% will escape.
Okay.
Go to the new place. So this is the thing which we discovered for the first time, that quorum sensing in a unison response can have reversible heterogeneity, and it's cheating. Reversible because that is very important. Because if you imagine another situation, if these cheaters always be there, then soon they will outnumber the performers. Because beneficial people will waste a lot of energy in producing their public goods, the goods which for the society.
Yeah.
And these cheaters will be doing freeloading for a long time. So freeloaders will naturally grow faster because they are not spending the energy, they will outnumber the performer and it will wipe out the colony, right? But nature plays a very fine switch between switching in of cheaters and non-cheaters. So this is called a reversibility. So in that way, it takes care of the double-edged sword. So one way it is allowing quorum sensing to perform social tasks. It is allowing to maintain some population of cheaters to do the job of the specialized job when the need comes. And it is also taking a hold on the cheaters so that they will not grow to wipe off the
whole.
So this is our main discovery, actually.
Yeah, that's a very interesting thing. So we need to move to a next question for which we need a very short answer. Is like this kind of study perhaps can help us in managing the problem of infection in food crops, right?
Yeah,
I mean, how do you think this application can come?
Yeah, so we have applied some of them. So what we found out that this, as I told you, that language molecule is very crucial.
Yeah.
So how you can confuse the pathogen? By either breaking the language or making the language in high amount, so that it will be like your sound system.
Okay.
So if you switch on the volume too high, then you will be not perform your task. Okay. So we have adopted the second approach. We have made transgenic plants which are able to produce the bacterial signaling molecule in high amount.
Okay.
Because now in those plants, when the bacteria enters, it will be fooled to think that we are too high in number, we should not go.
Okay.
This is one strategy. Okay, that we have published also. But since in India transgenic work is not that much of use, we have developed a non-transgenic approach also.
Okay.
In that we make that synthetic molecule, this signaling molecule in large amount in some other host or from that bacteria genetically modified and spray them on the plant.
Okay.
And now the plants are spiked up with the signaling molecule. And again it will do the same job. When the bacteria enters inside the plant, it will think we are too high in number, we should not attack. So this is what is called now coming up as a social medicine, like antibacterial, antisocial drugs or antisocial chemicals.
Very.
People in abroad also they are trying. In Pseudomonas aeruginosa, they are trying to fool the bacteria in terms of iron metabolism. We are also doing some work on iron metabolism, in the social iron metabolism. And this social microbiology is a big scenario in Europe. Many labs in Europe, they are now trying to attack the social system of the bacteria instead of attacking one target using antibiotic, where the resistance is very easy to get. But if you attack the social structure, everything is gone.
Very nice. I mean, we had a very wonderful conversation, and we got to know your work, the area of your research, and also how it can possibly benefit the future generation. Thank you for being with us in the show. Keep watching Eureka. We will come back with another interesting conversation next week, same time. Thank you.